individual's fitness, right? Offspring measure of fitness. According to Ricklefs, that's not enough. You can produce offspring, but it's the contribution of an individual's descendants. So it isn't until these individuals produce offspring that now this individual listed here has a fitness. In other words, I have 4 kids. Under these 2 definitions, and actually that one as well, I have a fitness, and that fitness would be 4 because I have 4 offspring. On the other hand, under Ricklef's definition, I don't have a fitness because my offspring do not yet have offspring. Does that make sense? All right. Okay, let's get back to HIV and see if we can incorporate some of this natural selection thinking and fitness thinking into how the virus is perceived by all of this. Okay? All right. This is to remind me to mention to you again that these antibodies that have been developed against a particular protein, in order to get this, the T cell has to pick up a little piece of the protein, present it to a B cell, and then the B cell begins to secrete these antibodies, right, to this original peptide that elicited this response. The protein that's on the surface of that virus, right, we know one of those, it's GP120. There's also that little piece here that's GP41. We know that that shape, if it's picked up by a T cell, presented to the proper B cell, we could get an antibody that recognized that protein, and that would allow the macrophages to destroy the viruses that are currently wandering around through the body. Okay? If, on the other hand, the shape of this protein changed and was no longer the shape recognized by this antibody, in order to get the immune system to recognize this slightly different protein, I would have to go back through this entire process again. Finding another protein, albeit slightly different from the original, elicit a new B cell to respond, producing a new antibody that would target this novel protein. All right. 3 very different plots. Let's just stick with the one at the top for right now. And let's look at our axes. The x-axis is the years since the patient became HIV positive. That's when we could first sense the RNA molecules that are associated with HIV in the blood. Right? We do blood tests, we pick up that RNA within that system. We know that we've got some RNA genome associated with HIV. On the y-axis is something called genetic distance. Okay? All right, so genetic distance is a measure of how different 2 sequences of nucleotides are. So, if I had one sequence of nucleotides, let's call it an RNA molecule, that's a sequence of nucleotides, and then I should have a particular sequence, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay? So let's just make it a short sequence so I can draw it on the whiteboard here. This is the particular sequence of RNA that I have associated with my HIV. Now, let's take a different HIV, look at its RNA molecule, and let's say along its sequence, it looks like this. Right, the way I've drawn it, these nucleotides at these 2 positions are different. Okay? They're different, and that would mean that 1, 2, 3, 4, 5, 6, 7, 8, 9 out of 10 nucleotides, there are 2 nucleotides different. That would be a 0.2 or 20% genetic difference. Okay? We're measuring how different 2 nucleotide sequences are. That's the genetic distance. How different are they? Okay? All right. Notice initially, by the time the individual became HIV positive, that's when we can first detect the RNA within the system. The virus has been around a while. It didn't just spring up here at time 0. It actually was present in the individual before this, but it took a while for enough viruses to be produced that we could detect it in the circulatory system. Is that okay? So at this point, there is a little bit of variation. So if genetic distance here is 0%, notice that the first data point after 0 is a little bit higher than 0%. That's because we have a lot of viruses already in the system. Why are these viruses already a little bit different from each other? The main culprit here is reverse transcriptase. Reverse transcriptase, if you recall, is not really good at making DNA from RNA. It makes all kinds of mistakes. And with these mistakes that are made, mutations are introduced into the RNA molecule. The mutation ends up in the DNA, but because the DNA is used to transcribe another RNA, the original RNA and the RNA produced through transcription will be a little bit different as mutations are accumulated. Is that okay? So every time reverse transcriptase does its thing, you get these novel mutations resulting in new pieces of RNA, and we can quantify how different these new pieces of RNA are from the other RNAs that exist within the population. So the HIV, because of reverse transcription, has a way to introduce genetic diversity into the population. It has a way to make individual viruses different from each other because of the mutations introduced through reverse transcription. Okay? All right, now, if you look at this line, it initially goes up like this. So this is differences accumulating in the molecule over time. That's right. And anything over time is a rate. So this is an increase in differences among individual RNA molecules that's occurring over time. The steeper the line, the faster these differences would be introduced into the population. The shallower the slope, the slower these mutations are introduced into the population. All right? Okay. Now, at this point, it looks like we're up around 0.075 on that chart. This means that at this point, any 2 random RNA molecules differ from each other in about 7.5 nucleotides right out of 100. That's the 7.5% distance that represents the genetic distance. 7.5% is 7.5 out of 100 nucleotides. I look at a 100 nucleotide sequence, 7.5 of those on average would be different among between any 2 individuals. Is that all right? Now, a point of clarification here. Well, 7.5% for a genetic distance, is that a lot or is it not a lot? right? Well, so we can ask, well, is this a lot of difference between these two viruses? So point of comparison, if I took your DNA, anybody's in the class, and I picked out a sequence of nucleotides to look at, and I picked another random individual anywhere else on the planet, and I looked at that same sequence, and I compared those two sequences to each other, what's the genetic distance? Right? Any two random DNA molecules will look at the sequences, what's my expectation for genetic distance? Or how different are people from each other genetically? Our genetic distance is about 1%. 1% for humans, 7.5% for these viruses at this point. What about if I took this same sequence of nucleotides in a human and compared it to the same sequence in a chimpanzee? Right? We should be pretty different from chimps, I would hope. Genetically, the genetic distance is about 4%. So over time, looks like over about 7 years, the viruses in this individual have accumulated more genetic distance in comparisons with other HIV viruses than we would expect to occur between humans and chimps, almost twice as much. So we have to ask then, well, how is this happening? How are we generating so much genetic distance among individual viruses so quickly, even to the point where we're dwarfing these differences between species? The virus is accumulating more mutations than I would expect, differences than I would expect between humans and chimps. Okay? All right, so the answer lies in this whole mechanism. If the RNA molecule, through reverse transcription, we introduce mutations, and then these mutations are then copied into the new RNA molecule. If the mutation occurs in a particular location, and this location is in a particular protein that's then recognized by the immune system, if I can change that sequence, I can change the protein. And if I change the protein enough, what used to be recognized by that antibody is no longer recognized. And I have to go back through the system again in order to find a new B cell that makes an antibody that recognizes this new protein. Right? In other words, the HIV population inside this individual that's HIV plus is evolving. This is what this is. This line here and its associated slope is telling us the rate of evolution of that virus. Right? More specifically, it's telling us how quickly individuals are becoming different from each other based on this model of reverse transcription. That's the rate at which the population is evolving. Is that okay? All right, now, consequences of this process. Well, my expectation is that if I plotted a specific HIV genotype, let's start down here with an individual that has a particular genotype. It infects the cell makes more copies of this genotype, plus some others that are different because of mutations that accumulate. My expectation is that the growth rate of this virus would be essentially exponential. Not indefinitely, because down here, when this genotype is rare, there aren't many individuals that share that genotype. But as the genotype infects more cells, the population of that viral genotype increases exponentially until some time when there's enough of it that by chance the immune system, the helper T cells, gets a hold of one of the proteins from this virus. Okay? The more common it is, the more likely the immune system stumbles on one of these proteins. And once the immune system stumbles on it, it begins to develop antibodies against this specific protein, and it should drive the number of individuals down. That's the immune system operating on that particular genotype of viruses. Now, other genotypes are being produced. These other genotypes are different enough that I have to establish a new antibody for it. So let's say at this point a new virus was established, and this virus then begins to grow exponentially until the immune system finds it and knocks it down. Other virus genotypes appear. They have to be knocked down as well. And each of these viruses, because of the proteins that we're altering the speed with which new viruses can be produced inside the cell. So if we think about the growth rate of each of these genotypes, so this would be viral genotype A, there should be a certain growth rate for this virus, and then there's also a viral genotype B growth rate, C, D, and however many others of these viral genotypes there are in the system. So I could have some of these that when they grow very slowly because it has problems having the cell make copies of itself. I could have other viruses that grow very quickly. They have a higher viral growth rate. Right? Now, in immunology and medicine, we talk about the ability of a virus and to grow in a population based on its growth rate. The growth rate relates directly to its virulence. How